Good evening, everyone, and welcome to the last lecture of the Berkeley City series and MSRI series, which is called, actually, It's Not on the Test. Let me introduce the lecturer of this evening, Dr. Phillip Sabes, and who's a professor of physiology at the University of California in San Francisco. I also learned that Professor Sabes did 2 undergraduate degrees, so one in physics and one in mathematics at Cambridge. And after that, changed a bit career and went into neurobiology. So he's also the director of the UCSF Swartz Center for Theoretical Neurobiology. His laboratory works toward understanding how the brain controls movement, and in particular, the role of sensory information and learning. His lab is also using this understanding, to develop brain-machine interfaces to help people with severe sensory and motor loss, such as people with spinal cord injury. He and his team have won awards for their work on the development of artificial, I practice that one, somatosensory feedback. So he's speaking tonight about the basic science and recent technical advance in this field. And it will also, I hope he's going to describe a bit of the work that they do in his lab.
First of all, I'd like to thank Berkeley City College and MSRI for inviting me. It's already been a lot of fun. So I'm going to tell you today about I'm going to give you a little bit of an overview about the state of brain-computer interfaces, and in particular, brain-computer interfaces for the restoration of movement and sensation. And I'll also be telling you as part of that about work that's going on in my lab. Before I get started, I just want to acknowledge the members of my lab who have done all the work that I'm going to tell you about. In particular, those who are in bold. Most of them I'll mention again as I go along in the talk. These are our current and recently former lab members who are primarily responsible for the work that I'll be telling you about that did come from my lab. Also want to mention the funding agencies that have funded this work, and in particular, DARPA, the Defense Advanced Research Projects Agency, that has taken a really big interest in this area, as you'll hear more later, and funded most of the work that I'm going to tell you about. Okay, so I'm going to tell you about brain-computer interfaces to restore motor function. And I'm going to define what I mean by brain-computer interfaces in a minute. But before I do that, I just want to tell you what's the clinical need? Why are we doing this work? So there are a variety of patient populations that have need of assistive devices. First, there's amputees. And the incidence of amputees in the US is well over 500,000. Many of those are lower limb, but about 40,000 are upper limb. And I'm going to be talking mostly about upper limb devices today, although a lot of the technology does generalize. In addition, there's spinal cord injury, almost 250,000 people. The numbers on these things are surprisingly fuzzy. The epidemiology is hard to come by. But this is on the low end of estimates. There are other estimates, say from the Christopher Reeve Foundation, that are almost 4 times as high. And then other forms of paralysis, including stroke, multiple sclerosis, ALS, etc. And the incidences there are perhaps 4 times greater than the incidences of spinal cord injury. So all in all, we're talking about well over 1 million people who have need of assistive devices, and this is the target that we're looking at. So when I say brain-computer interface, what do I mean? Well, what I'm going to be talking about today is specifically motor brain-computer interfaces. So what I mean there is a prosthetic or an assistive device that can help somebody with daily tasks of living, an interface to that device that allows the individual to give some input into the assistive device, and I'll make all of these clear as we go along. And then some kind of controller, and this is principally where the math comes in. It's about how do you take signals from a device, from an interface, and use that to control a device. Okay. So let's start with looking at the devices themselves, the physical devices that can help people. So as I mentioned earlier, DARPA has a long-standing interest and has invested a great deal of money in the development of prosthetics, neural prosthetics in particular, that is, prosthetic devices that are controlled by either the brain or the peripheral nervous system. And in particular, DARPA has funded the development of two really nice, very high-tech prosthetic upper limbs. These are devices that are meant to weigh roughly the weight of a typical adult male limb. They have the full degrees of freedom, the full movability of a natural limb. They're also sensorized, so they have the ability to have touch in them. They're still experimental tools. They're not available yet in the clinic, but there's already a lot of success. And I'll show you a video. This is from a 60 Minutes piece on, hopefully the audio is going to work here. This is from a 60 Minutes piece on the DECA arm. And the DECA arm comes from Dean Kamen. It's his company. Some of you may know Dean Kamen because he's also the person that developed the Segway, among other technical marvels. Okay, so I think the video speaks for itself.
Okay, Kamen asked Fred Downs, the VA official in charge of prosthetics, to take off the hook he'd been wearing for 40 years and give the new arm a try. The arm is controlled by flexing the shoulder and pressing buttons built into his shoes, almost as if he's typing with his toes.
Ball of the foot, outside of the foot, and then toes.
After practicing for 10 hours, Downs showed us what he could do.
Now I use my toes to grasp this.
So I hope you appreciate it. I think this is pretty remarkable, right? So this is somebody who's been getting by with a very useful but much more rudimentary device, a hook, which is still very commonly used. And here he is, after only 10 hours of use, able to pick up a Coke bottle with this device. So that's really fantastic. But it still leaves a little bit to be desired, right? It's a slow interface. You can't use your limb while you're walking because it's controlled by the foot. And so this idea of having other body parts control the assistive device is useful, and it's how most people who have devices like this interact with their devices, but we'd like to do better. There are other devices that people are working on. The prosthetic arm that I just showed you obviously is ideal for amputees, but there are other devices that are perhaps more applicable to cases of paralysis, like exoskeletons. This is a video of an upper limb exoskeleton that's being developed by Jacob Rosen's group at UC Santa Cruz. Also devices such as wheelchairs and computer interfaces that I'm sure many of you have seen in a variety of places. For example, the kinds of things that Stephen Hawking uses. So all of these are devices that are available. Some of them are still research, some of them are readily available, and we're interested in how it is that one can control these in a better way. Okay, so I told you about devices, and now I want to talk about interface. So how is it, and in particular, I want to talk about brain-computer interfaces. So how is it that you can get signals directly out of the brain that it would allow you to control some of these devices? Where do you start? Well, it turns out we know where to start from neuroscience. There's an area of the brain called primary motor cortex. The cortex, or cerebral cortex, is the outermost part of the brain. It's also the last in sort of phylogenetic development, so it's sort of, in some ways, the most sophisticated part of the brain. And there's a region of this part called primary motor cortex, and this has been the principal target for brain-computer interfaces. And the reason for that is because this is the part of the brain that, at least principally, controls volitional movement. So if you want to move, if you want to do something, it's motor cortex that allows you to do that in conjunction with other brain areas. But motor cortex receives input from all these other brain areas that are involved in the sensory and cognitive control of movement. And then it projects down to the spinal cord and actually controls the spinal cord and the muscles that allow the limb or the trunk to move. So this is an obvious place to look to get signals out that you might use to control one of these devices. How would you access those signals? Well, again, there are devices that are available. This thing here it looks like a little bit like a pincushion. It's a little silicone array, 4 millimeters by 4 millimeters, that has 100 electrodes on it. And it's fairly compact, and it was developed at the University of Utah in collaboration with Brown. And each of these electrodes, so it has these electrodes, and it also connects to this connector that's externalized. So right now this will sit, and you'll see some images of this in a moment, but it sits on the head, and then this part hooks up to some wires, so that goes through the skin percutaneously. And these electrodes can be used both to record electrical signals, and I'll tell you about that first, but they also can be used to electrically stimulate the brain, and I'll tell you about that later in the talk. This very device, well, maybe not this physical one, but the same device has been used in animal models, as I will tell you about later, but also in human patients. And you'll see some videos of that in a moment. So this device is right now FDA approved for clinical trials, and it is almost all of the studies that are currently, the clinical trials that are currently ongoing, that are brain-computer interfaces are using this device. Okay, so there are devices that you might want to control. There are ways to get signals out of the brain. So what do you do? How do you link these two? And this is a really interesting problem. It borders between basic science and engineering, and it's also where the fun math comes in. Okay, so I'm going to tell you about the brain-computer control of external devices. So the first thing you have to know a little bit is about what happens when you stick electrodes like this into the brain. So again, if we go to motor cortex, what I'm going to show you here is a trace. This is an electrical recording that we've made in my lab from a monkey using the exact same device that I just showed you. So the same electrode arrays, we use those in our animals as well. And in a moment, I'm going to start this video. And what's going to happen is there's going to be a blue line that moves along. And you'll hear sound, hopefully. And you'll hear some rumbling, which is kind of background noise. And then you'll hear some pops. And those pops are these events, these big amplitude events. And then what I've done is every time one of these big amplitude events happens, I'm going to trigger to plot over here a 4 millisecond window around that pop. Okay? So this is a 15-second video, and I'm giving you 4 millisecond snippets. And you'll see why in a moment. So you're listening to a real brain. Okay? This is what you get to listen to if you're in my lab. And there's this sound, this zzz, almost like a motorcycle revving. And that's times when this neuron that we're recording from has more of these events, and then there are times when there are less. And these events, you'll notice, are highly stereotyped. They all look almost exactly the same. And the reason for that is because this is an action potential, or what we call a spike. So the way that neurons communicate with each other is not the way, say, for example, a telephone communicates through the line with an analog signal that can go up and down. Instead, it communicates with discrete binary events. There is or there is not a spike, and when a neuron spikes, it conveys information to downstream neurons. And we can record these. They're the basic elements of computation in neural circuits. And they're also the driving force of the outflow from motor cortex down to the spinal cord. So we can record these things, and often when we do these sorts of experiments in the lab, what we're mostly looking at is just these spikes and the rate at which they occur. There is other signal in this, but I'm not going to talk about it today. I'm going to focus principally on the spikes. Okay, so how do you go from this sort of faster rate, lower rate, that sort of thing, to knowing something about movement? And this is the problem of inferring behavior from spike rates, which is what we call the problem of neural decoding, looking at neurons and decoding what the animal is or wants to do. And this problem borrows heavily from statistics, machine learning, and engineering. So this is the kind of thing that we have animals do in my lab. We have them sit at a table and make reaching movements, center out reaching movements like this. And if you look at the patterns of movements that you get when you do this, these are each of these different colors is a movement to a different target. There's some variability, so when the monkey's moving to the same target, there's some variability in what he does, but he can reach these targets. Now, if we look at the rate of the firing of these spikes, the rate that these events occur, in a motor cortical neuron, so a neuron from motor cortex, as the monkey's doing something like this. There are times, again, where the rate would be low, like here, so the neural firing rate is low, the spikes per second is low, and there are times when the neural firing rate is very fast, like there. And what I've done here is I've plotted that firing rate on a per movement basis, so an average of about some window during the movement, as a function of the very initial velocity component. So in some cases, like here, the monkey's moving outwards. We're going to call this 90 degrees outward. And when the monkey does that, that neuron fires a lot. And in some other trials, he's moving inward at minus 90 degrees. And in that case, this cell doesn't fire very much at all. So what you can see is that the firing rate of this neuron correlates very nicely with the velocity that the monkey is moving with. In this case, it's actually it's a linear correlation. So when there's literally a correlation in the mathematical sense between roughly the sagittal direction, that's the outward direction that the monkey's reaching, and firing rate. When he reaches out this direction, high firing, down this rate, low firing. Okay, that's nice. So how might we use that in the context of a brain-machine interface? Well, when we record, remember I told you we have 100 electrodes, so we're recording from many neurons at the same time. And you can imagine that this neuron tells you a little bit about the sagittal direction. You might have other neurons that tell you about other directions. And if somehow you could pool them all together, you might get a pretty good estimate of what the monkey's doing. And in fact, that works. So we're going to use these correlations across a group of neurons. We're going to train up what we call a decoder, and then we can predict going forward what the monkey's doing just by looking at the neurons. And in this case, even a very, very simple schematic of the simplest thing you could do with this sort of thing, which is linear regression, which I'm sure many of you have learned about in a math or a stats class. Even simple linear regression works. And in this case, what that basically means is that there's some true velocity, and we're going to estimate that velocity. Whenever you see a little hat on a variable in my hands, that means it's an estimate. There's a true velocity out there, but we're estimating that velocity based on firing rates. And so the way linear regression works here is there's a bunch of neurons, each one has a firing rate, and there's some parameter that we have to fit. That's the process of regression, and we get a good estimate that way. And you can think about this intuitively as each neuron, when it fires a spike, is sort of voting for some direction of velocity. I think the animal's moving in that direction, in essence. And we're averaging across those. So these parameters that we fit during regression are the direction that each cell is voting for, in essence. Okay, so this works. And what I mean by it works is if we look here at, for each of these movements, we just plot the very initial component of the velocity. So that's, velocity, remember, is a magnitude and a direction. So this is a vector. So in these, for example, the monkey reaches with pretty much a constant speed off in this direction. This is what the monkey actually did, and then we can predict how well he did on future trials with 79 neurons. And we do a pretty good job. We capture about 62% of the variance in this. So this isn't a perfect match, but we're getting more than half the variance. We're getting the gross structure. So you can imagine that if you're just trying to get a cursor to move on the screen, that maybe this might be good enough to get you started. Now, is this good or is it bad? 79 neurons, how do you think about that? Well, 79 neurons seems like it's sort of a lot. If you're a neuroscientist who grew up, a few decades ago, people would record from one neuron at a time. That was the technology. So 79 is pretty good. On the other hand, that little 4x4 millimeter chip that we put in there has something like 1.5 million neurons beneath it. So 79 is a pretty small number. So in fact, this is pretty good given the small number of neurons that we're recording from. Okay, now, in practice, linear regression isn't really good enough for what we want. Neurons are noisy, they're variable from trial to trial, and as I said, we're only recording from a very small set of these neurons. And there are other problems as well. Neural circuits actually aren't stationary. That's a mathematical term, which means have the same statistics over time. They move around, their statistics change over time, and so simple linear regression that wouldn't necessarily capture that. So there are ways to build better mathematical models of neural and behavioral data that allow you to perform better. And the state of the art actually is something that I'm just going to give you a flavor of what that looks like. And it uses two pieces of information. It uses the neuron behavioral correlations. That's what I just told you about. That's the linear regression part. But it also uses knowledge not about neurons, but about the behavior that the animal or the person normally makes. So it learns about the statistics of that behavior independent of the neural activity. So for example, it doesn't happen that the monkey starts moving here and then instantaneously finds his hand over here. Movements are smooth. It knows about that. And the solution for how you combine these two things in some special cases is actually very well known. It's an extremely widely used algorithm from statistics, engineering, machine learning. Everybody knows about this in a variety of fields that do processing of dynamic data, and it's called the Kalman filter. And what the Kalman filter basically says is that your estimate is a combination of a prediction component, given only knowing where I just was, knowing what my velocity and my position just were, what should I expect to see in the future? It doesn't take into account the neural data. And then another component, which is like a correction to that uses the neural data. And this is the state of the art. Okay, so what does it mean to be state of the art? Now I'm going to show you some more videos. You made it through the math. Here's some good videos. So this is a video from Brown University and Mass General Hospital. Lee Hochberg and John Donahue jointly working on this. And what you're going to see is a patient with this, again, the same array that we have that I showed you before and that's used in animal studies. And this is a tetraplegic patient, and you'll see what happens. So her goal here, as you can see, is to pick this up and take a sip. You notice there was a little fade out there, so I've edited this video down. Now, how can you not smile when you look at that, right? I mean, this is amazing, right? Here's a woman who, cannot move, has not given herself a glass of water to drink in years, and with the help of this little chip and a lot of computer code, she's able to reach, grab this thing, and bring it to her mouth. So that's fantastic. On the other hand, it is quite slow. It's fantastic, I agree. You can clap. That said, it is slow. I edited the video down. It takes considerably longer. So this is great, but you can imagine something that's better. So this, by the way, this was using a Kalman filter algorithm. This is based on work from Krishna Shenoy's lab at Stanford University. And there's a group here at Berkeley, Jose Carmena, who does very similar kinds of work. And what they're doing is adding sort of bells and whistles to the Kalman filter. And the key idea is to sort of retrain it as it goes to make it update on the fly. And this is actually monkey work. So you're going to see a monkey. It'll be obvious what he's supposed to do. He's supposed to reach to the little colored dots. And he's using this sort of bells and whistles version of the Kalman filter. So this is pretty fast. This is impressive. There's a monkey thinking. There's no arm movement. He's thinking about moving around these obstacles and moving to these targets. So that's pretty impressive. Now, that said, they're not smooth movements. They don't look like normal human or monkey movements. But it's fast. It's impressive. This is what you can do just by sort of improving that algorithm a bit. Okay, one more example. This is the most recent of the examples. And this one actually uses an algorithm that's kind of basically the linear regression algorithm that I told you about. Except, again, there's some bells and whistles, mostly having to do with how the human subject was trained. There was an extensive training regime that involved constraints and then relaxation of constraints. Another tetraplegic patient, and you don't see the patient here. The patient is controlling this robot arm. The patient's sitting somewhere like over where you guys are, and controlling this robot arm. And what the task is to pick it up and drop the contents into this measuring cup. So again, I mean, these are really lovely demonstrations. Okay, so impressive performance. It's great advance, yet it's still not at the level of natural control. And so the question is, what's limiting the performance here? You might think, well, it's the neural interface, and you're only recording from on the order of 100 neurons. There are millions of neurons. You need more neurons. And we do need more neurons. And there are other reasons why we need better interfaces, and I'll talk about them briefly at the end. But I actually think that may not be the major problem here. It's part of the problem, but it may not be the major problem. Another thing you might say is, well, these control algorithms, the things that control engineers have known about for decades, surely you could do better than that. Well, control engineers are pretty smart, maybe not. And people have been working really, really hard on this. I actually don't think that the principal problem here is that the control algorithms aren't good enough, which isn't to say that you can't do better. People are thinking of really creative ways of incorporating biological knowledge. Krishna Shenoy, in fact, is doing some really great things along this line. But I don't think that's the principal problem here. What I think the principal problem is that subjects are relying purely on visual feedback. Now, It might seem like that's fine. You know, you go, you pick up your coffee cup, you pick up your cup of water, you walk through the world. We're very visual creatures. You think probably that mostly what you do in the world is you rely on vision. So that shouldn't be a big problem. But I'm here to tell you it is, okay, and here's why. If you look at patients, there are a small number of patients who lack what we call somatosensation. That's the sense of perception of one's own body. There are two versions of that. You'll hear about that in a moment. If you look at patients who lack somatosensation, you'll see that it's devastating, and it has major effects on motor control. So that's what I'm going to talk about now. I'm going to tell you about proprioception and its role in movement control. This is a fantastic old documentary from the BBC, which I'll admit is available on YouTube. So if you're interested, you can find it. So I'm going to show you a little snippet of it that introduces what we mean when we say proprioception. By the way, the American spelling is an O here, proprioception.
Charles Fried has lost control of his body. A rare virus has ravaged his nervous system, leaving him with no sense of touch below the neck and unable to sense how his limbs are moving. Charles is not paralyzed, but he has lost his proprioception, the hidden sixth sense that tells the brain where the body is in space. Proprioception is so basic to our experience of being in the world that most of us are unaware that we have it.
So that's amazing. I mean, if you saw this person out on the street, you would think that he had some just debilitating motor deficit. But in fact, his motor system is intact. The problem is his sensory pathways. Now, as the narrator said, proprioception is something that many people have never even heard of, and they don't think of it as something that's important to them. But actually, it's very easily appreciated. So if you ask yourself, can I close my eyes and touch my fingertips together? How is it that you're able to do that? Your arms are starting over here, yet you're able to match them together. In order to do that, you must have some ability to know where your limbs are as you're moving them through the space. And that's proprioception. So it's easily appreciated, and it's sorely missed when it's gone. Okay, now, let me show you some other examples of what happens. That's really debilitating. That's a patient who's pretty much lost all somatosensation. But even if you have patients who are just lacking or heavily impaired somatosensation of the upper limb, you still get deficits. So here's an example. This is a great study by Bob Sainberg and Claude Guez, an early study in the 90s, where they asked subjects to do a simple task, to take their hand and pretend they were slicing a loaf of bread, something like this. It might seem like a strange thing to do, but the reason they did it is because that reversal, that quickly moving and coming back, is something that requires a fair amount of force and actually requires well-timed coordination between the shoulder and the elbow joints to get that right. And if you look at normal subjects, they have no problem with this. You can do it with your eyes closed. It's not a problem. It's an easy task. But if you look at patients who are lacking proprioception, what you see is that they can do it, but it's clearly quite uncoordinated. The timing of the joints, which is normally quite precise and stereotyped in normal subjects, is off, and as a result, they make movements that are kind of wiggly and squiggly. In fact, they look a lot like the kinds of movements that you saw in that monkey video from the Chenoy lab. They're good, they're fast, they're not too bad, but there's something wrong. They're uncoordinated. And so proprioception, we know, is actually really critical for interjoint coordination. This is a quote from another early paper in the 80s from the Rothwell group, where they were looking at these kinds of simple laboratory tasks and showing that without proprioception, most of these simple things like reaching from one point to another, the patients could do. They could execute the tasks. But despite his success, this is one patient, despite his success with these laboratory tasks, his hands were relatively useless to him in daily life. He was unable to grasp a pen and write. He was unable to fasten his shirt buttons or to hold a cup in one hand. So that's clearly a very devastating sort of disability. Now, this deficit is both due to proprioception, knowing where your limbs are in space, and due to the lack of tactile feedback. That's the other kind of somatosensation. That's the one that you're more familiar with, the sense of touch. I'm going to show you a really great demonstration of a video of what happens when you lack touch. And this comes from Roland Johansson, who's done a lot of the really great work on the role of tactile feedback in dexterous hand movements. So first I'm going to show you this video, and this is just a young woman who's going to strike a match with one hand. So this is not a terribly difficult task. Okay, so she does it. She does it in about 8 seconds. Now, in between these two videos, what Dr. Hansen did is he injected lidocaine into the fingertips of this woman so that she could no longer feel, she had no sense of touch on her fingertips. And watch what happens now. So she's having trouble picking the thing up. And then once she has it, she's having trouble controlling it and holding on to it. Again, there's nothing wrong with her motor system. This is purely a problem of lack of somatosensation. So she gets it, but it takes a lot longer. Okay. So I hope you can appreciate that somatosensation is really critical for motor control. And we argue that brain-computer interfaces without somatosensation will never achieve the kind of natural fluid movement that you'd like them to achieve. So having somatosensory feedback would improve control. There's another thing it would do also. It would improve the sense of embodiment. So we're used to our body belonging to us. Our limb feels like it's part of us. And if you can feel an assistive device, if you can feel a robotic limb that you're holding onto, it will make that limb seem like it's more like you're part of your body, which makes the experience better, which will increase the rate at which they're used and adopted. So we think that that's another reason why it's important to have somatosensory feedback. So how can we achieve it? Okay, so I want to tell you a little bit about what's out there in the world for artificial sensory feedback, and then I'll tell you about two approaches to delivering artificial somatosensation. So when you talk about brain interfaces, or really any artificial somatosensory device, the technology that's principally available these days is electrical microstimulation. So what does that mean? That means that you're going to take one of those electrodes that I just showed you and pass current through the electrode. Instead of recording electrical voltages, you're going to pass current through the electrode, and you're going to drive neural activity. So by passing current, it goes into the tissue, and that causes these spikes to happen. The best example of this, and the earliest example, is a cochlear implant. So these are devices where, where you can pass an electrode through into the inner ear, into the cochlea, and it spirals along the cochlea. And the cochlea has a really great property, which is that the cells along the cochlea are all tuned to frequencies in this topographic way. So there's a map from high frequency to low frequency as you move along the cochlea. And it has to do with the physics of the fluid in the cochlea and the hair cells and so on. But what this means is that if you put an electrode in here and you stimulate, based on where you are along the spiral, you'll kind of know what tone the person will hear, either a high-pitched tone or a low-pitched tone. So these are out there. They work. Many people use them, and they work very well for speech, for example. Maybe not, to hear a symphony at the level that you or I might, but they're really great for communication. Okay. More recently, people have been developing retinal prostheses that use exactly the same idea. Put a little chip of an electrode onto the retina, put a little camera, maybe mounted on glasses, and play patterns of electrical stimulation into the eye to drive the nerve in cases where the photoreceptors aren't working. Now, these are both out there. There are FDA trials now for these. So these work, but the question is, how do they work? There are two main things that's going on here. The first is that they're creating patterns of activity that are what I call biomimetic. That is, they mimic the natural patterns of activity that you see in the cochlea or in the retina of the eye. And this works because these peripheral organs have an organization, a topographic organization. The location of the visual scene maps to the location on the retina, and the frequency or the tone of a sound maps to the location on the cochlea. So can you use this for the case of artificial proprioception? So now I'm going to tell you about some recent work on biomimetic somatosensory feedback. This is not from my lab. So I told you about motor cortex. Well, right next to motor cortex, a little bit further back, a little more posterior in the brain, is an area called somatosensory cortex. And some of you may be familiar with this picture that originally came from Penfield, who did the original stimulation studies, where he would stimulate and ask people what they felt, where in their body they felt a tingling sensation. And what this shows is that, say, for example, the face and head are represented with a lot of real estate in somatosensory cortex, the hand as well, and maybe other body parts like the trunk less so. And the idea is that this map of the body on the tissue, on the cortical surface, is what we refer to as a homunculus. So there's this body map, this body image on the brain, the somatosensory homunculus. So what you're supposed to get from this is that the sensory cortex is topographically organized, just like the cochlea and the retina. So maybe you can go in and stimulate in somatosensory cortex and use that to create artificial somatosensation. And indeed, this is work underway. I'm going to tell you about some work from Sliman Ben Smaya's lab at University of Chicago. What he did was he had monkeys place their hands on a table, and he stimulated their fingertips with a little mechanical device. So he indented a little bit the fingertip, just a little poke. And if you do that, what you find is that if you record neural activity in somatosensory cortex, so we had two little arrays sitting in somatosensory cortex, that different electrodes responded best to stimulation, to tactile touch of different parts of the hand. And in fact, there was a topographic organization. So these fingers are more represented here, the palm of the hand more over here, and so on. Okay, well, that's nice. And then he showed, so what he did after recording is he performed what we call intracortical microstimulation, or I'm going to use the acronym ICMS for much of the rest of the talk. That means passing a little bit of current into the neural tissue and driving maybe like 100 neurons to have an action potential. And if you do that, what you find is that monkeys can detect both skin indentation, of course, but they can also detect that electrical stimulation. And they can even use it, they can even discriminate different amplitudes. So if I press harder versus lighter, you can feel that, of course. And they can also detect if there's a greater or smaller current pulse that was delivered. So that's kind of cool. Furthermore, monkeys can detect differences in location. They can detect which finger was stimulated, either mechanically or electrically. So, for example, electrical stimulation of two fingers that are far apart, the monkey pretty much got that right 100% of the time. But if the fingers were closer together, they weren't 100%. And these solid lines here are distinguishing two mechanical stimuli, same finger, or more to the left or more to the right. These textured lines are data from when the monkey was asked to compare mechanical stimulation on one digit to electrical stimulation on an electrode that represents another digit. And what you can see is that they were able to do the task not quite as well as they're able to distinguish two mechanical stimuli, but still fairly well. So this works. You can get a monkey to sort of feel something like that one of their fingers being touched if you electrically stimulate in this cortical area. Again, really cool. The question is, how do you get from that to the kind of real-time feedback that you need, especially proprioceptive feedback that you need to drive movement? Now, I'm going to argue that there are limitations to this biomimetic approach of trying to reproduce, take advantage of the topography and reproduce natural patterns of activity. The first is that sensory homunculus that I showed you, it's not really right. It's right at a very coarse level. It's not that Penfield got his data wrong. He didn't. It's just that when you go to a finer scale, the kind of scale of the hundreds of neurons that you excite when you stimulate with an electrode, those neurons can all have different responses. They can represent different parts of the body. And that's particularly true for proprioceptive feedback, even more so than for tactile feedback. So that means that even if you knew the right pattern of activity across these, it would be difficult to sort of recreate that pattern because the electrical stimulation is a sort of coarse tool. Furthermore, the way that experiment that I just told you about was done is first you touch the hand and you make that map, and then you take advantage of that map. But what do you do in a patient that has spinal cord injury, for example, where there is no longer somatosensation coming from the periphery, or an amputee who no longer has that limb to touch? So there are problems with this approach. So we took a very different approach. It's a complementary approach, but it's a very different approach, which is not to focus on recreating natural patterns of activity, but rather to focus on the brain's natural ability to learn. So we know that the circuits for sensation and the circuits for movement are highly adaptive. And there are lots of examples that I could tell you about. I don't have time today about all of them, although I'd love to. I'm going to focus on one example, which is multisensory integration. So I'll define what that means, and I'll tell you briefly at the end the role of learning. Okay. So what does the brain do when it gets information from two different sensory modalities? that tell it about the same thing. You could imagine maybe it just pays attention to one or the other. It keeps around both estimates, and there's some maybe dissonance that you hear one thing and you feel another. Let me start with a particular example of what happens when you have vision and audition telling you about the same thing. So here's a demo. Now, what I want you to do is to look at this video and listen and tell me what you hear.
Ba 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 ba.
So unfortunately, the timing's a little bit off in the video, but what did you guys hear? I hear fa, did I hear va? So va, so I'm hearing, who thinks va? Who thinks fa? Who thinks ba? All right, the timing was a little bit off. So I'm going to go with those of you who said va. Okay? So one of the things about sensory integration, I'm going to tell you more about this in a minute, is that timing is actually really critical. You can't have a lag between the two signals, or else the brain figures that out. Unfortunately, that was true. So let me try replaying it one more time, and let's see what happens.
Ba 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 ba.
Still off.
Ba 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 ba.
Okay, for those of you who heard va, what I'd like you to do now is listen again and close your eyes.
Ba 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 ba.
What did you hear now? Ba, clearly, unambiguous, right? There's no doubt that was ba. Pretty striking if you heard it right the first time. Now let me show you one more thing. I'm going to turn off the audio, and I'm going to play the video one more time, and tell me what you see. You can yell it out anytime. Fa, fa, right? I think most people see fa. Some people might see fa, but most people see fa. Okay, so except for the timing snafu, what normally happens with this video is that when you have just vision, you see fa. And when you have just audition, it's very unambiguously ba. But when you combine them, you get something in between, which is va. And in fact, this is what the brain does. Now, auditory people and speech people sometimes say that vision is disrupting audition. Audition was the right thing and vision disrupts it. But that's not how I think about the problem. I think about the problem that the brain is looking for consensus. So the brain is getting two signals, they're both valid, and it's looking for a middle ground. So it sees fa, it hears ba, and then with the percept that you get is va in between. And this is typically the rule. This is what happens when the brain is presented with conflicting stimuli from two modalities. And this is true for vision and proprioception as well. So say, for example, that your hand is actually here, you feel it here, but you see it over here. That's easy to do in the laboratory. We can do that, say, with prism goggles or with a virtual reality setup or something like that. And if you do that, what happens? Well, what would you do if you were a brain and you were told that my hand's here by vision and my hand's here by here by vision and here by proprioception? What would you do? Well, here's a sensible thing to do. Closing your eyes is a sensible thing to do. But sometimes closing your eyes only gets you deeper in trouble. You could ignore one modality, but that's actually not what the brain does. Another thing you could do is you could say, well, look, I know what to do when I get two pieces of data, right? I average them. That's what you do when you get two pieces of data. So I'm going to take these two estimates, and notice I've got a bunch of hats here again. There's some true location X, and I've got an estimate that derives from vision, an estimate that derives from proprioception, and I'm just going to take the average. And it turns out that's a sensible thing to do. It makes sense. People do that sort of thing all the time. And there's a reason that you do that sort of thing all the time. It's because averaging reduces variance. So if this thing has some known variance, say sigma squared, and this thing has some known variance, sigma squared. And in this picture, what these little blurs are supposed to represent some uncertainty or variability of the true location given the sensory signal that I have. Then by combining them, you get a 1 over square root of 2 improvement in variance, a reduction in variance. And that's why this yellow circle is smaller. So you're better at estimating when you combine two signals than when you only use one alone. And this is generally true. Now, in fact, there's no reason why vision and proprioception should have the same variance. So you can generalize this problem to the case where they have different variances. So for example, maybe proprioception is more uncertain or there's more noise involved in it than vision. And you might think that what you should do in that case is not throw proprioception away. It's not a terrible signal. If it were really, really noisy, maybe you throw it away. But if it's only a little more noisy, maybe you should weight them somehow, but weight vision more, and vice versa. And it turns out that is the right thing to do in a statistical sense. You can ask, what's the right way to combine these two signals that minimizes the variance of the estimate? And the answer is to weight the two and to weight them inversely proportional to their variance. So if this guy has twice the variance of this, then it's weighted half as much as this. And that's what this says. And if you do that, then the combined variance, this yellow guy, is reduced. It's this equation here, but this equation is necessarily lower than either of these two. Now, if one of these is infinite, if there's no information, if you have infinite variability, then it'll just be equal to the other one. Okay? All right. So this is the mathematically smart thing to do, and sometimes we're smart. So we and animals, this is what we do. When confronted with two sensory signals, we combine them in a statistically optimal fashion, at least at first order, pretty much across the board. There have been many, many studies looking at this, including, by the way, by Marty Banks, who's one of the seminal studies on this, who's on the faculty here at Berkeley in ophthalmology. Okay, so we know that this is true, and you might think that this is sort of a weird thing, that you have to go to the lab to get this kind of weird state where these two sensors disagree, but actually this is the normal state. Real sensory signals are noisy, so at any given moment, what you get from two sensory modalities won't be perfectly the same. And also, sensory signals can change over time. For example, the predominant way that you know about where your limb is in space is through these things called muscle spindles, which are sensors that live in your muscle. So if you go and you work out, you might change the tone of the muscle a little bit, and that sense can drift. So these things change over time, which can cause a discrepancy. And so, in fact, what's going on is that these two signals are constantly being recalibrated. If they get out of calibration, the brain brings them back together. It adapts the way that it interprets the same sensory signal to bring them back together. And we've demonstrated this in the lab. Many others have. This is an ongoing process. You're constantly relearning how to use your sensory signals. Okay, but that's what that was meant to say, that when they disagree over time, they get brought back together, they get recalibrated. This system is also, it has to be learned. You're not born with being able to map two sensory modalities perfectly. It's learned during development. And you can ask, how does this all happen? What's the mechanism by which the brain is able to do this feat of bringing these sensory signals together in an optimal way, adaptively? Well, we don't really know, but in my lab, one of the things that we do is we build models of this sort of thing. And we recently published a paper, Joe Macon and Matt Fellows, two postdocs in my lab, published what I think, at least, is a really great paper that shows that the brain can, in principle, we used a neural network model that was based on some ideas from machine learning, that are basically models of how you build statistical models of the world around you. And what it showed was that a simple neural network can learn to do multisensory integration. It can learn to do it in this statistically optimal way. And the only thing it needs is that those two signals correlate in space and time together. So if you have vision and proprioception, and they're doing the same thing, I'm looking at my hand, then the brain can automatically combine them. Okay, so that got us thinking. If that's true, then maybe the brain can use a signal that it knows about, like vision, to bootstrap the use of an entirely novel signal, like an artificial somatosensory signal. So at last, I'm getting back to artificial somatosensation. So a learning-based approach is what we call our approach. I'm going to tell you about this last experiment, and then I'll be done. This is very recent work. We just submitted the paper on this. The person who's really done this project is Maria Detterlot, a fantastic graduate student, a bioengineering graduate student. This is a joint UCSF-UC Berkeley graduate program that she's in. And Joey O'Doherty, a postdoc, who's been involved in this project, but is also taking this to the next step, which I'll tell you about at the end. Okay, so the question is signal correlation enough to drive sensory integration? Can we use that to get a learning-based approach to artificial somatosensation? So here's what we did. First, we trained the monkey on kind of a strange task. This is the kind of thing that behavioral neuroscientists like me will do. They come up with tasks like this. We asked a monkey to make reaches from one point to another. I've already showed you data like that. That's pretty straightforward. Except he couldn't actually see the targets. Instead, what he saw was a bunch of dots that randomly moved on the screen. You'll see some examples of this in a moment. If the dots moved in this direction, it told him that he needed to move in that direction. If the dots moved fast, it said move far. If the dots move slow, it says you're almost there, you don't have to move very far. So he would see that at the beginning of the trial. There was this preparatory period. We'd see that, plan a movement, then there's a go cue, and he makes a movement, and then that signal updates as he goes so that he can use it as feedback in real time. Now, this is a nice design because it allows us to change the reliability or the variability of the signal easily. If all the dots are moving together coherently, it's very easy to tell the direction of the flow. If, for example, half of the dots are moving in random directions and the other half are moving coherently, you can still tell, but it's a little bit harder. And then, of course, we can go all the way to where all the dots are moving in random directions, and there's no directional signal at all anymore. Okay, so we train the monkey to do that. That's the baseline task. And then what we do is we combine this visual signal with an ICMS signal, with stimulation. And I'll tell you about what that is in a minute, but it's multi-electrode stimulation of somatosensory cortex in a pattern that's entirely non-biomimetic. The monkey has never experienced this sensation before in his life. But it's going to correlate moment by moment with vision. And our hope is that this is enough to train the animal to learn to interpret that signal. And then we're also going to test, can he do that now after that training with just the electrical stimulation? Okay, so this is a video. I'm sorry, it's kind of a bad quality video, but it's just a hand, I think it was a cell phone actually, of a monitor in the lab that's mirroring what the monkey's seeing as he's reaching. And what you're going to see is, this is the monkey's view. There's a start target, so he knows he has to move here, and he can see where his hand is because there's a spot of light. When he gets to the start target, it'll go away, and a bunch of dots will start flowing, and he'll know what that means, and you'll see what he does. So he made the right reach, he saw those dots. That was a really good one. This one, he has to correct a little bit. It's a challenging task, but he can do it. This was actually fairly early in his training. Okay, so monkeys are very good at this sort of thing. People are very good at this sort of thing. If anybody wants, after I suppose I have a demo on my laptop, you can try it out. I can't do the stimulation part, which, boy, I wish I could do myself. It'd be really interesting to see how that feels. That would solve a lot of problems. But anyway, the visual task is fairly straightforward. Okay, so how do you quantify this behavior? So we can look at, for example, there are lots of metrics that we use, and they all give the same answer. But what I'm going to focus on right now is the variability of the initial angle. So we're going to look at, given that he's supposed to reach here, how variable is that reach? And you maybe, those of you who are more mathematically savvy may see why I'm doing that, because this is essentially a proxy for how well he can estimate that initial angle that he's supposed to be reaching to. If he really knows where he's supposed to reach, this variability should be low. And if it's hard to parse that signal, then the variability will be high. And indeed, that's what we see in this metric. When the coherence is 100%, all the dots are moving together. It's very easy to tell what's happening. The variance of the initial angle is low. And as the coherence goes down, so the dot field becomes harder and harder to discern where the direction of the flow is, the variability of the initial angle goes way up. So that's pretty straightforward. Okay, good. So again, this is one measure, but everything I'm going to show you is the same for all the measures we've looked at. Okay, so now what? Now we're going to stimulate the brain. This is the exciting part. So how are we going to do this? We're going to stimulate with a pattern. We're going to put one of these electrodes in somatosensory cortex, just like the other experiment I told you about. And we're going to stimulate with a pattern that's arbitrary, that has nothing to do with real neural signals, but it's going to map to vision. And here's how we do it. It's going to look a little bit familiar. It's like the dots. Each electrode, there are 8 that we're going to stimulate on, that we know the monkey can detect stimulation on. We've already tested that. When those pulses, those little electrical pulses happen fast, that means the monkey has a far to reach. And when they happen slowly, that means he's almost there. So the overall rate tells him distance. And then the difference between the rates of the different electrodes tells him direction. And I'll very quickly tell you how that works. If you notice, it doesn't really matter. It's just that the difference between electrodes tells direction. And the way that happens is each electrode is arbitrarily assigned, having nothing to do with the normal activity there. One of the 8 vectors here that I'm showing. And so, for example, if the monkey has to reach in, say, around the 180 or 135 degrees, so in this direction, then these 2 electrodes should be having the fastest rate of pulses, and they are. And then these electrodes over here should have the slowest rate of pulses, and they are. And what happens in this particular case is the monkey goes and then overshoots a little bit. So now these guys at the end of the trial are the lowest, and these are the highest because he needs to come back in the opposite direction. But of course, the overall rate is lower because he's close to the target. Okay, so this is what the monkeys were exposed to. Now, early in training, for training, what we did is we combined vision and ICMS. So this is just a video of one trial. So what's happening is the monkey reaches to the start. There's a delay period where he sees the flow, he experiences these pulses, and then boom, he goes and he reaches. And he's pretty good at this. This is an easy task for him. All right? So he trains with this sort of thing, combined stimulation and vision. Now notice, he could ignore this. There's no reason why he has to pay attention to the stimulation. He's already been trained to do the task with vision, right? So he can ignore it. But those 2 signals correlate moment by moment. And I've already told you that if you have 2 signals and you average them or you weight them, then you'll do better. So if the brain knows what it's doing, if it's looking to do as best it can, it'll combine them. But it doesn't have to. And this is what happens after training. This is a task where there's, oh, no, that's the same video. This is what happens after training. So here, he's going to go to the start position, and then he's going to move, but there's no visual feedback. All he has is the stimulation in his cortex, and he starts off in the right direction, and he goes there. Notice, I'm showing you the target here in dotted lines, so you know what he's supposed to be doing. He can't see that, of course. All he gets is this electrical stimulation. So this is, this was a really, when we saw this, we were obviously quite thrilled. Okay, so what does the performance look like if we quantify it? So this is, again, the data that I've already shown you for vision alone, and this is what you get for this stimulation, this ICMS, intracortical microstimulation, alone. And what you can see is that performance is comparable to 25% of the dots moving in the same direction. It's a little cloudy, but you can see the direction. You can really tell. So this monkey knows what he's doing when he gets that stimulation. So it's pretty good when you get ICMS alone. But remember, the whole point of this I was telling you is that if you have two signals, you should be able to combine them and do even better. So does he? Well let's look. So here, at 100% coherence, when vision's very good, then vision's very good, and it doesn't really help to add the stimulation, and in fact, vision and combined are about as good. Here, at 0% coherence, vision's not really telling you anything useful, and in fact, these two are not statistically significantly different. The critical cases are here, where vision and stimulation have about the same reliability, and there, indeed, you get a good deal of benefit from having both cues. So that's nice, but is it the best? Is he combining these the best way that he could? Well, how would we ask that? Again, remember, we've already shown that natural stimuli, the brain will combine in this minimum variance way. So we can go back to the same equations that I showed you. We know what these variances are because the performance that we get with the individual modalities is a proxy for these numbers. So we can simply apply this equation and ask, does the performance on the combined task do what it should do if the monkey's making optimal use of these two cues? So again, these are the data, and this is the model prediction. It's spot on. And there are other ways that we can assay this, and indeed it does look like the monkey is combining these cues in a statistically optimal fashion, which again is the way that he would do it if they were two natural stimuli. Okay, so let me summarize the results of this experiment and then give you a little bit of an overview, and then I'll stop. So I've shown you that a learning-based approach to artificial somatosensory feedback works by even with arbitrary mapping of spatial variables. The monkey's still able to learn, and he does that because vision and ICMS are correlated. Not only does he learn to use ICMS alone, but he does it in a very natural way. These experiments, by the way, really were motivated by computational models and looking at human behavior. Just the ICMS performance alone sets a new benchmark for what can be done with stimulation in somatosensory cortex in terms of getting performance with that kind of feedback. So we're quite pleased with these results. Okay, now more generally, going back to sort of the broader scope of the talk, I think the videos that I showed you earlier, I think you'll agree that truly remarkable progress has been made in the development of brain-computer interfaces for the restoration of motor function. In particular, there have been some fantastic assistive devices that have been built, and the algorithms for VMI control have gotten quite sophisticated.